Thank you very much for giving me this opportunity to present our work, which indeed is a collaboration with the group of Frank Johannes that you will hear just after me. So we will split our talk into two parts. I'm going to do the molecular bit, and Frank is actually a quantitative geneticist, and he's going to take you into the intricacies of transgenerational epigenetic inheritance and how we can try to address issues about this sort of bizarre buzzword of these days, transgenerational epigenetic inheritance. So let's start with a set of questions that I would like to address today, or that we are jointly addressing, Frank and I, in this project. So first, we want to understand to which extent perturbation of delimethylation patterns can be inherited across generations in plants, and we believe that plants are particularly prone to this kind of inheritance of epigenetic changes from one generation to the next. And then, of course, what are the long-term consequences, both molecular and phenotypic, the consequences of this methylation perturbation, of this delimethylation perturbation in plants. And again, we believe that perhaps changes in epigenetic states may provide actually a new source, or a source which has been unexplored so far, of heritable phenotypic variation. And this is basically what we want to address in our project. And so, of course, Arabidopsis is a superb model to carry out such studies, because we can perturb delimethylation patterns readily by just using a whole bunch of mutants, some of which have been described by Steve this morning, and which are interestingly both viable and fertile. So we can do genetics with them, and that's something that plants are uniquely gifted with. If you do the same type of experiments in a mouse, you get embryonic lethality most of the time, and therefore you can't do any work on transgenerational effects, or at least you can't do them with the readiness that you can do in plants. Now, we're going to focus on one particular perturbator of delimethylation patterns, which is DDM1, that again Steve introduced this morning. So DDM1 was first isolated as a recessive mutation which had an impact on global methylation levels throughout the genome of Arabidopsis. And basically, in a DDM1 mutant background, what you get is about 70% reduction of global DNA methylation. And just as soon as the mutation was isolated, people who studied it, namely the group of Eric Richard and Roy Martinson and Tetsuji Kakutani in particular, they noticed that the changes in delimethylation that were induced by DDM1 could be inherited independent of the DDM1 mutation itself. So basically, you could segregate out the DDM1 mutation by making a cross to a wild-type plant, and then you will keep this hypomethylated state. And you could see that through classical Southern blot analysis of digested DNA using enzymes that are sensitive or insensitive to DNA methylation, or you could do it genome-wide using a very coarse technique, but which is actually quite sensitive, which is HPLC. And you can measure the amount of 5-methylcytosine, and what you can see is that you have in the F1 an intermediate value between wild-type and DDM1. And because DDM1 is a recessive mutation, this implies that somehow you inherited your hypomethylated state. And so we went on with the group of Roy Martinson to show that in DDM1, what you see is actually a major restructuring of the epigenome. So basically, heterochromatin, which is mostly composed of repeat sequences, transposon, but other type of repeat sequences, is usually in a heavily methylated state in wild type. As well, associated with this heavy delimethylation, you have dimethylation of lysine 9. Again, Steve mentioned that this morning. And then in a DDM1 mutant background, what you see is an almost complete erasure of this delimethylation, which is centered on repeat sequences. And you have now a gain of this active chromatin mark, which is dimethylation of lysine 4 of its own H3, that you can't see in a wild type. So now what was heterochromatic now looks completely euchromatic across the entire domain that we probed at that time, which was about 1.5 megabase. So we knew that DDM1 was a very important epigenetic regulator, so to speak, and that if you compromise its activity, you will actually induce a whole series or suite of epigenetic defects or chromatin defects. So we used this, and we knew quite a few other things about DDM1, the fact that you could indeed inherit not only changes in demethylation states, but you could inherit phenotypes in a DD1 mutant background. And two examples are shown here. One, this is a mutation which was originally isolated by a group in Holland. And it turned out that FWA, which was actually induced by EMS mutagenesis, is a hypomethylated allele of this particular gene with a transposon upstream of it. And so we went on to show that this particular methylated sequence in a DD1 mutant backgrounds get hypomethylated, and this hypomethylation then is transmitted to the progeny independent of the DD1 mutation. So this is how you can now get this heritable dominant FWA mutation. And here we have something that does occur in DDM1 as in many other methylation mutants, which is basically that you mobilize transposon because you reactivate them transcriptionally. You not only lose DNA methylation of the transposons, but you actually gain expression, which is indicated here in red. In DDM1, as you can see, all of these transposons are now heavily transcribed when they were silent in wild type. So this reactivation of transposons, in some cases, not systematically, but in some cases, does lead to transposon reactivation and mobilization. And this is one example, CLAM, which was actually isolated by the group of Tetsuji Kutani. This time, it's a sort of an obvious DNA sequence alteration. You have a transposon sort of landing into a gene and then, of course, knocking out that gene and leading to a recessive mutant phenotype that you see here, the CLAM phenotype. So that's a sort of picture on which we decided to explore this issue of epigenetic variation across generations. So what we did was a very simple scheme, or the idea was very simple, is to start with two plants that were identical except for the DD1 mutation. So these are two plants that derive from the Columbia accession, but one carries a DD1 mutation. And of course, because it carries a DD1 mutation, now you have the epigenome, which is dramatically altered with these hypomethylated loci, plus a few transposition events, perhaps, and perhaps other type of DNA sequence alterations. But we believe that these are minimal compared to the massive amount of epigenetic restructuring that occurs in a DD1 mutant background. And then we cross this plant to make an F1, and then back cross it to wild type to get rid of the DD1 mutation, which I've indicated here. And so by doing that, now you create a series of individuals which are all the same genome, except for the few, perhaps, DNA sequence polymorphisms that segregate or that distinguish wild type and DDM1. So you have essentially the same genomes here. So you have isogenic lines, but that differ from each other through their epigenome. The DDM1-derived epigenetic state versus the wild-type-derived epigenetic state. And then the issue is, what happened to this alteration that you induced in one generation? So it's a transient perturbation of DNA methylation through one generation that then you segregate out. What happened to this perturbation 6 or 7 generations in a wild-type background for DNA methylation functions, including DDM1? So basically, do you carry through these epigenetic alterations, or are they reverted and gone back to wild type? So this is what we wanted to set out, and we wanted to do it in a framework of a quantitative genetics approach. That was important for many reasons, because first we wanted to explore genome-wide the impact of these epigenetic changes that DDM1 does induce, and second, we wanted to see the phenotypic effect of such epigenetic alterations in a context of quantitative genetics, of quantitative traits, which we know are the primary targets of selection in nature. So to do this, and as I said, Frank is going to elaborate on the quantitative genetics approach, but here what we set out to do was to take 100 of our epiRILs that we generated here. So we generated 500 such lines, and we decided to epigenotype 100 of them. So of course, nowadays we could do it by using the technology called bisulfite sequencing, whole genome bisulfite sequencing, but that's still a bit expensive. So instead, what we turned to was to use a technique of MEDIP chip, MEDIP standing for methyl DNA immunoprecipitation. So we immunoprecipitate DNA after fragmenting it with an antibody that specifically recognizes 5-methylcytosine, and we co-hybridize it after labeling into different colors on a tiling microarray, which covers the entire Arabidopsis genome at about 165 nucleotide resolution. And so the first thing that we wanted to do, of course, was to make sure that wherever we extracted our DNA from, we will get the same delimethylation profile, so that there was no issue with dynamics of delimethylation during plant development. So we know that there is some dynamics, but most of these dynamics has to do with probably the reproductive phase, and therefore here what we are dealing with is methylation profiles in shoot versus roots, and as you can see, they are extremely similar, if not identical, given that there is always a bit of noise in these genome-wide approaches. So here we are confident that wherever we will take our DNA from, but of course we took it consistently from the same organ or same tissues at the same stage, we will get a consistent profile of DNA methylation. And this is what the signal looks like. So when you do the ratio of immunoprecipitate to total DNA, there is, of course, a large fraction of the genome which is not methylated. We know that because there are very few repeats in the Arabidopsis genome, and therefore we know that because DNA methylation is mostly targeted to repeat sequences, we will have only a limited amount of methylation. And so this is consistent with all the measurements that were done before, HPLC, BS sequencing, whole genome BS sequencing. So most of the genome is unmethylated. And then there is two categories that we really wanted to distinguish, which is the highly methylated DNA and the intermediate demethylation level. So we did that through using hidden Markov models, where you take the correlations between probes in order to derive a confident call about methylation status of a given probe. So here we distinguish three types of sequences: the one that are unmethylated in the genome, the one that are with intermediate methylation level, and the one that are fully methylated. And this is what it looks like now when you compare wild type to DDM1 using this hidden Markov model. So basically, as I told you again, most of the genome is not methylated and is therefore unchanged in DDM1. It's therefore unchanged. This is actually maybe not a surprise, but something that we needed to confirm. And what you can see that actually there are not too many sequences that do get hypomethylated in DDM1. So the one that were heavily methylated in wild type and become completely unmethylated in DDM1, or the one that were fully methylated in wild type and that became intermediate methylated in DDM1. So what these sequences that lose methylation resemble or look like? Well, most of them correspond to transposon sequences, and again, this is consistent with everything we know about the function of the primary function of delimethylation in plant genomes, and secondly, what we know about DDM1, which seems to specifically target repeat sequences and does not touch much gene body methylation. So here, the three categories that are the two main categories that lose methylation, MI and MU, as you can see in terms of annotation, correspond mostly to transposon sequences. So all of that is sort of confirmatory to what we knew already. But now what's interesting is because we are exploring 100 progeny of these DDM1-induced alterations, so we can really now start to look at the stability of these hypomethylated states that were induced by DDM1 in a quantitative manner, using quantitative genetics approaches, but also having the sufficient numbers of individuals to say with confidence whether a sequence has retained a methylation profile or that was this hypomethylated methylation profile that was induced in DDM1, or whether it was reverted to wild type. And what you can see is that actually there is a very neat distinction between two classes of sequences. The one that we call MU, so they were fully methylated in wild type and lost all or most methylation in DDM1. They are the gray ones. And then the ones that are in purple here, MI. So they have, again, full methylation in wild type, but they only lose partially their methylation in DDM1. And as you can see, we expect, if we take a random sample of 100 epi-RILs, that they should segregate 25% at any given locus, given our crossing scheme where we have an F1 and then back cross to wild type. So at a given location, if you take 100 epi-RILs, you should have 75 individuals with wild type methylation patterns and 25 with DDM1 methylation patterns, if things are inherited in a Mendelian fashion. So this is quite true for the MU. It sort of follows a binomial distribution, but not for the MI type of sequences. So the one that lose only partially their methylation in DDM1. So have a tendency to regain wild-type methylation in the progeny. So here we already can distinguish two types of sequences, the one that seem to be stably inherited in the hypomethylated states when they have been perturbed by DDM1, and the one that have this capacity to reacquire or to revert to wild-type methylation. But what you see is that reversion is not fully penetrant. So not all of the lines are at 100%. We have this sort of gradient of reversion. So basically, you have for a given sequence, maybe 80 or 90% of your individuals that will have reverted, but not 100%. So you have variable penetrance in terms of reversion. And again, I think this is something that we can only address because of the large number of lines that we looked at. So where do these two types of different sequences distribute along the chromosomes? Well, as you can see, there is nothing that distinguishes them in terms of chromosomal location. Of course, as we expect, most repeats are in pericentromeric regions, but both the reverting and the stable hypomethylated alleles will be found in this region, and the same along the chromosome arms. So there is no distinction there as to what will make a sequence more reverting than another one based on chromosomal localization. Now, we needed to validate a little bit more this delimitation analysis based on MEDIP, and here this is a collaboration which actually involved also Steve Jacobson, who made some bisulfite sequencing libraries for us and that we sequenced then for 6 epiRILs. And what you can see, we have a very good correlation actually for a given epiRIL between the methylation call that we make based on MEDIP. So here is wild type, a set of probes. These are individual probes, so they're all wild type here. Here is intermediate, and here is DDM1. And as you can see, the bisulfite signal actually follows very well the MEDIP signal. So we can trust our data quite well. And what's important then is that you can use this information to determine the haplotype, the epi-haplotype, sorry, I should say, of this particular chromosome in this particular epiRIL. Because here what you see is that you have mostly wild type or intermediate probes in terms of methylation level. You have very, very few hypomethylated probes. So this is telling you, at least in terms of probability, that here you are dealing with an interval that must have come from the wild type parent. And here you have a breakpoint, a recombination breakpoint, and now you have the rest of the chromosome which must have derived from the DDM1 parent. So this is extremely useful, and Frank is going to elaborate on this, because based on these haplotypes, then of course you can create recombination maps and start to map perhaps phenotypic variation, heritable phenotypic variation to stable delimethylation variation. So here I just want to focus now back on this reversion, which is not always fully penetrant. And what are the characteristics of the sequences that here don't seem to follow this sort of very simple, naive assumption that if you lose methylation, there is no way you can regain it. You lose it forever, and therefore you get these stable hypomethylated alleles, epi-alleles. So we had actually done some work on that a few years ago, which was published, and we came to the conclusion that there was one major discriminant between what we call remethylatable or reverting epi-alleles from the one that are stably maintained in a hypomethylated state, is that this class, which is non-reverting, is not targeted by the RDDM pathway that we heard of this morning. RNA-directed methylation, if it works there, is very, very inefficient, because once you lose this methylation, so these sequences, even when you bring back the DDM1 function, have no ability to regain delimethylation, and that's the only way you can establish methylation is through this RDDM pathway. So which, of course, begs the question, how do you get methylated in the first place if RDDM is so inefficient? So that's something for which we have no answer at this stage. And here we have a set of sequences that are revertant. And yes, what distinguishes them is that here they are associated with an abundance of small RNAs, which is the signature of this RDDM pathway, both in wild type and in DDM1. So basically, even in a mutant background, you still carry this RDDM function, which, by the way, explain this MI category that I talked about. I being that we have in DDM1 intermediate methylation level. We don't lose all methylation. And this intermediate methylation that we have in DDM1 associated with these reverting sequences has to do with the RDDM pathway kicking in constantly in this mutant background. And of course, if you bring back now this DDM1 sort of master orchestrator of maintenance methylation, then you can fully reestablish, you can reacquire full wild type methylation. And then there is a minority of sequences in the genome which are constantly targeted by the RDDM pathway, but not by the maintenance machinery. So they are insensitive to DDM1, and indeed we have actually confirmed that through our genome-wide approach, that we have a few sequences that are M in wild type and stay M in DDM1, and stay M in all of the epiRILs. Another thing that we observed was that this process of reacquiring or reverting is actually a slow process. It doesn't occur in just one go. And that's actually why initially people didn't see it when they analyzed their F1, between a wild type and DDM1 parent, and they had this intermediate methylation level. It's because there was no remethylation taking place in the first generation. It starts in the second generation, just like what David talked about this morning, and then it sort of goes into increments. So every generation, you acquire a little more from what you started with, and you maintain that throughout vegetative life. And then the next generation, you acquire a little more again. And this, we know, fits extremely well with all of the activities of the RDDM pathway at the time of reproduction. Again, David mentioned that this morning. So what can we learn more? By now, I should have said that all of these analyses that we have used to come up with this model and this discrimination between reverting and non-reverting epi-alleles were based on just a bunch of sequences, about 50 or 100 sequences that we actually analyzed by hand through various means, PCR, etc., etc. Here we have now this genome-wide view of 100 progeny. So what can we learn from looking at that at a population scale? And the first thing that surprised us is that I told you that density of small RNAs or abundance of small RNAs was a very good correlator or indicator of capacity to reacquire DNA methylation, to revert to wild type once you have lost methylation in DDM1. Well, it's actually not the best correlation that we can get. We get a better correlation if we now count the number of distinct small RNA sequences that match a sequence. But the best predictor of reversion for us is a mean hit number of these small RNAs. So this may be a bit complicated for you, but so you have to realize that first, we're dealing with repeat sequences, so we have to put all kind of filters to make sure that we are not confounding different transposon copies with each other. So we have to do first a filter where we only query transposon sequences that are unique or for which the sequences are not ambiguous. So we know that we can rule out cross-hybridization in our MADP experiment, and we know that there should be not too many copies exactly the same. There should be no more than one copy exactly the same elsewhere in the genome. But now, for the small RNAs, we have these small RNAs that are 24 nucleotide long, and we ask for a perfect match. And of course, they can match to many places if they come from repeated sequences. Of course, what we don't know if they come from all of the repeat sequences simultaneously, or one of these repeat sequences produce a whole lot. But what is clear is that irrespective of the ways that you produce your small RNAs, it's not so much the total quantity, the density of small RNAs that you produce that matters, but the fact that you produce it from sequences that have similarity elsewhere in the genome. And that immediately suggests that you are dealing with repeat sequences that are sort of young, that sort of moved not long ago, have kept sequence similarity between them. Divergence, nucleotide divergence, was not too extreme yet. And of course, this is where also it makes sense to have the RDDM most active. It's when the repeat sequences, the transposons, have just landed somewhere, started to increase in copy number, and there is high sequence similarity between copies. And so maybe there is a chromatin state there in this particular DNA a chromatin state at these particular copies that sort of makes them extremely sensitive to the action of RDDM, not through making a lot of small RNAs, but instead of making them on a chromatin, talking to a chromatin which is extremely recipient to this information. So maybe it's, I lost you here, but there are all kinds of arguments that suggest that this is not the bucket load of small RNAs that matters, but more the quality of the small RNAs, where they come from, and what type of sequences they interact with. So there is definitely a chromatin component as a recipient end for RDDM to be efficient. So then we wanted to go from individual probes to a sort of more global picture where we now consider domains because, of course, all of these analyses carry some noise with them. So here we want to define, for a given stretch of sequence, whether domains that could be reverting domains or non-reverting domains. And doing this approach with a very simple filtering scheme, we have about 2,500 reverting domains in the genome and about 1,000 non-reverting domains. So, by the way, 1,000 non-reverting domains, that's 1,000 potential stable epi-alleles for phenotypic variation later on. And so when we look at what they correspond to, they correspond, of course, to only a tiny amount of the entire complement of T sequences that we have in the genome, because we have applied so many filtering schemes. The T's that are transposable elements that are too similar to each other, we will have thrown away, etc., etc. But still, this is informative. Whenever we have a domain, we usually have only one T fragment. So this is immediately suggesting that a given T will have a given behavior. It is either remethylatable, reverting, or non-reverting. You don't have bits of the sequence that will be reverting and bits of the sequence that will not be reverting. So the targeting seems to be a function of that particular copy, not of particular sequences within that copy. And so you don't have this sort of mixed behavior. And this is actually illustrated perhaps more easily here, where we put the total number of transposon sequences for which we could ascertain the quality of reversion or non-reversion. And as you can see, there's a category where for a given annotation you have both a part of the sequence that might be reverting and in terms of methylation status, and another part that is non-reverting is really a minority compared to the other two categories. And of course, the predominant category is the one that is reverting. So these sequences have still this ability to reacquire DNA methylation if they lose it transiently through whatever means. But you have different behaviors for different elements. So gypsy class, most of the elements are reverting. But now if you take to the line class of elements, it's just the opposite trend. So here what you have, it's this one. So you have as many, if not more, non-reverting line elements in the genome than you have that are reverting. And we actually know a few examples of line copies in the genome that may have an impact on gene expression nearby. So these are all potential suppliers of epimutability in the genome. And now, I told you, maybe in a very convoluted and not very clear way, I realize that what matters is not so much the amount of small RNAs that you produce in order to get efficient reversion. Is perhaps more the fact that these small RNAs come from young sequences. And so here we have further evidence that this is indeed one of the main parameters that govern this efficiency of remethylation. What we did is very simple. We took all of the non-reverting sequences and the ones that are reverting, and we compare them to their reference sequence. When you annotate a transposon, you do it in reference to a given transposon which is deposited in red base update or whatever. And so what you see here is that there is more similarity, of course, among reverting copies than you have among non-reverting copies. But here we are worried because these reference sequences, sometimes they are constructed through a very odd way, and so we don't know what they actually mean, whether it could be that one reference sequence is a very, very old sequence, and therefore everything that is similar to this particular sequence would be reflecting a very old age as opposed to a young set of insertions. So here what we do is carry out a similarity comparisons, this time by taking a random set of 2 sequences, comparing them, and we do that reiteratively for many different families, and again we get the same result. So when you compare now 2 members of a given family to each other, if they are non-remethylable, their level of dissimilarity is greater than if they are reverting. So this is further confirmation that sequence similarity, age of DNA sequence of these repeat sequences is probably one of the major determinants of reversion. And here I'm just illustrating a few of the examples that we found of the different classes of sequences. So this is an ateline 1,6 element, quite large, 6 kb long, but with a bit of insertion here, so it doesn't count in our annotation. And you can really see that in this particular epiRIL, the profile is exactly as in wild type in terms of methylation. You have as little methylation as you had in wild type, or actually you may have lost a bit of it. But you still have small RNAs. But maybe they're not at high hit number. And here you have another copy of a line element, which is, again, a bit obscure with this interruption in the middle. But here you can see that the methylation level in this particular epi-RIL, we know that this segment comes derived from DDM1 based on this haplotype that we can derive based on the entire set of probes along a chromosome. So we know that this particular sequence sits on a DDM1 derived chromosome, and yet its methylation profile is exactly like wild type. And again, it's not very clear to see what could be the determinant of this sequence being reverting when the previous one is not reverting. And here we have one example of a composite sequence where a bit of it, but actually it's not a bit of it, so this has to do with annotation, so we have to, again, be very careful about that. We annotated this sequence as being both in some part reverting and in other parts non-reverting. And the non-reverting part is actually a mixture of sequences that flank it at the end. Now, so much for the distinction between these two types of epi-allelic variants that we can induce in Arabidopsis. One class that will be stable for many generations. I should have said that initially, but our epi-RILs have been now propagated for 6 or 7 generations, some of them 14 or 15 generations, and we still see this epi-allele in a stable hypomethylated state. So we do have things that have a stability which is perhaps not as strong as DNA sequence variation stability of DNA mutants, but still strong enough to be carried for maybe 20, 30, or 40 generations, and therefore that could be subject to selection. And then we have the other category, which will regain progressively over 3, 4, 5, 6, or 7 generations their full wild-type methylation. And now there is another aspect, of course, of all of this epigenetic variation, is that it does induce structural variation in the genome. And here we started to explore the space of this sort of structural variation, which is generated by this transient perturbation of delimitation patterns by DDM1. So what we did was we took the same 100 epi-RILs that were subject to epigenotyping through this MEDIP technique, and now we're sequencing them, 30x coverage using parent sequencing, like again what Steve described this morning, where you try to see whether the 2 pairs of a segment map in the same position in your reference genome, or one now has to be put elsewhere in the genome. That will be an indication that something has transposed. And this is the result that we get. It's not perfect, actually still a work in progress. What I should say that most of the the SNPs, we have SNPs. Most of these SNPs are just a difference between our Columbia and the reference Columbia. So we have very, very few SNPs that seem to distinguish our DDM1 from wild type. So we can confirm that most of the genome is identical between our two initial parents. But we have a few transposons that jumped. And some had jumped in the DDM1 parent, but others had jumped in the epi-RILs as we as we propagate these lines with some sequences that remain in the hypomethylated states and therefore have retained the potential perhaps to be transposing. But as you can see, we don't have a big explosion of the genome. It's still very limited. So if, and Frank is going to elaborate on that, but if you still, in this population, if you start to see heritable phenotypic variation, then you are in a very good position to actually assign it to delimethylation variation and not DNA sequence variation, because we have very, very, very limited DNA sequence variation in this context. So, and with this, I need to conclude, and by saying that transient perturbation of delimethylation over repeat elements leads to 2 main classes of hypomethylated alleles, epi-alleles in Arabidopsis, one stable and one unstable. Age of T sequence seems to be the major determinant of what will determine the efficiency with which you can reacquire DNA methylation through the RDDM pathway. Stable epi-alleles can be used to determine with great certainty the parental origin of any interval along the chromosome, so we can now use them for deriving recombination maps based on epi-haplotype and not DNA sequence variation. And we believe that these stable epi-alleles can potentially provide a rich source of heritable phenotypic variation. And again, Frank, I hope will convince you that this is the case. Thank you.